stepping right outside the doors. Which brings me to the first etiquette rule. If you guys would put your phones on silent, that would be wonderful. The other thing is, this whole area is technically considered behind the scenes, and the reason for that is that there's no big wall separating us from the dolphins. So this makes for a pretty awesome classroom and a beautiful view. But it also means that we are required by law to keep our eyes on you all and make sure that nobody decides to step out of here and go for a swim. So if you do step outside for a phone call or a restroom break, one of our very well-intentioned staff or volunteers may kind of poke our head out the door, and I swear we're not following you, but we're just making sure our dolphins are well cared for and that we are obeying the law. That's all for rules here. You guys, this is your space. This is our Marineland classroom that we use for various things, including our summer camps and our school field trips and our lectures and staff training. So settle in and enjoy yourselves. A couple of things before I pass it over to Kenneth. This program, you may be here because you are a longtime fan of Marineland, and in which case, thank you for loving us and for being here today. And you may be here because you are a longtime fan of the GTM Research Reserve, and you've never been to Marineland, in which case, welcome. We are so glad that you are here today. You may be here because you are a big fan of UNF, or you love mussels, or you hate invasive species. Regardless, we're really glad that you are here today. Mostly this is about cooperative programs and bringing people together who, whatever your area of interest, you care about this planet, you care about the oceans, and whatever inspires you and whatever motivates you, it got you here today. And for that, we are grateful. So get to know each other, because you all do come from different backgrounds. Get to know us and enjoy yourselves. All right, and with that, I will hand it over to Ken with the Research Reserve.
Hi, yes. So my name is Kenneth Rayner. I'm the Education Director for the GTM Research Reserve, or our full name is the Guana-Talamado Matanzas National Estuarine Research Reserve. Very long name. So, what I wanted to mention about this lecture is we are very excited to be able to co-host this lecture with Marineland Dolphin Adventures, just because it does bring our entities together and working together and producing this lecture series, and we're very glad to have it. And what's great about this lecture series, what we focused on this year with the series is on conservation. That's our main theme, is conservation. Because it's great to have that knowledge and awareness, but then taking that knowledge and awareness and actually doing behaviors and actions with it, it's even better. So we're targeting conservation both on the local level and regional level, and some of our talks have been on the international level. So we encourage you guys, it's every third Tuesday of the month at 10 a.m. here is our Marineland Lecture Series. Now, with that being said, it does require an RSVP, which is a free RSVP, through our booking system, our online system. Just noting that we don't open the registration for next month until you guys got to give us at least a good 3 hours to get back to the office to turn that button back on. So just give us 3 hours after this, and we will hopefully have it ready for you guys to register next month. I think that's, one other thing, just housekeeping things, is when you get home, you will have an email generated from the online system thanking you so much for attending, but there's also will be a link to a survey on there that's a quick online survey. If you could please just fill that out for us, because this program was actually designed from those survey responses. You guys tell us what you want to hear and what you want to know about. So that really helps shape what we bring to you guys. So if you could just fill that out for us, that would be wonderful. So on to the show. Oh, one more lesson. Sorry, housekeeping. So much housekeeping. Is we do broadcast this live. It is actually, I'm on YouTube Live right now, so the world is seeing me live. So this is something we're trying out new to bring our lecture series beyond the classroom and into whoever wants to watch it. We're just trying this out now. So this video and this presentation will be available once you get home. You're welcome to share it with whoever you want. Noting that we are still experimenting with this system. We are not Facebook IT geniuses and things like that. So we are playing around and trying to get it to work. So, but that's something that we're trying out. But just an FYI that it is live right now on the internet. So you guys are internet. The camera's this way though, not that way. So just letting you guys know. I heard that little dingo. All right. Well, I would like to welcome our guest speaker today. It is Dr. Matt Gilks from University of North Florida. And Dr. Gilks' research focuses on looking at invasive species and speciation, but also conservation genetics. And Dr. Gilks is here to tell us the story about the green mussel. So without further ado.
Thank you. First, thanks for the invitation. Thanks everybody for coming out. You guys are a little bit different of an age class than I'm used to at the university, but that's probably a good thing, actually. So anyway, thanks for coming out. I'm going to talk to you a little bit about some of the work that I've been doing. I've been at UNF for about 13 and a half years, and I've done work. I do a lot of work in Europe, but since I've been here, really over the last 10 years, a lot of my work is focused on some of the problems in Northeast Florida, and I'm going to tell you about some of that here. Primarily, the reason I chose this topic is because a lot of the work was actually done in GTM NER. So hopefully you'll be able to see a lot of the, I don't have a whole lot of pictures, but you'll see some of the results of that work for those of you that are familiar with the NER. And so the first thing that I'm going to do is, is talk to you a little bit about the terminology that we use, just so that you'll be familiar with everything that I say, or at least some of the things. Because a lot of the words, scientists, we love words, but the problem is a lot of times people use these different words for the same thing. So if you notice this, we've got four terms up here. Introduced species, non-native species, non-indigenous species, exotic species. All of those basically mean the same thing. Those are all terms that are used interchangeably to basically describe a species that is currently living outside of its normal range or its historical range. Now you'll also hear the term invasive species. That term, however, typically has a little bit different meaning. So not only is an invasive species an organism that is living outside of its historical range, but it also has it's causing some sort of problem, either economically or ecologically. So that's usually what we reserve it for, are organisms that are causing some sort of a problem in the local area. And here we go. Technology, right? All right, so when we talk about the ways that organisms are introduced, we have to also realize that they can be introduced through a number of different means, and in some cases they're on purpose, right? So we've got purposeful introductions. So this is where humans introduce a species to a new area by design. So we take it with us for a specific reason. So you think about most of the agricultural plants that we have. Most agricultural plants in the United States, citrus, other fruit trees, basically everything except for like corn and peppers essentially came from Eurasia and other parts of the world. So these are organisms that we have taken with us on purpose to provide food, or stocking fish that we like to fish for, things like that. So I've got a picture of largemouth bass for those of you that fish do any freshwater fishing. Those are native to the southeastern part of the United States, and we've basically introduced them throughout the country to 49 out of the 50 states, everyone except for Alaska. They've been introduced to Spain, to Japan, all kinds of places, and we do it on purpose as a sports fishery. There are also natural introductions. So natural introductions are basically the result of infrequent events where species get blown off course in some cases, in the case of birds or something like that, where they end up establishing a population where they previously didn't exist. Some of the greatest examples of this are volcanic oceanic islands like the Hawaiian Islands, the Galapagos Islands. Those islands haven't been there forever. Volcanoes erupt and basically the lava forms these new islands out in the middle of the ocean. Nothing lives there at first until something occupies it. So all the organisms that get there essentially are living outside of their historical range. Therefore, we call them introduced, but they weren't introduced by us. It was a natural event. So these are just some of the examples of finches on the Galapagos Islands that I've got there. And then you've got the ones that I'm going to talk about primarily today is an unintentional introduction. So these are human-mediated again. Humans have introduced organisms to a new area, but it's by accident. So usually these are hitchhikers of some kind. They come along with you when you take a plane somewhere. So snakes, these are a brown snake that occupies a lot, that lives in a lot of, they've been introduced to a lot of the Hawaiian Islands and things like that, are decimating bird populations. And these guys hitchhike on the wheels of planes, of airplanes. So when planes go from one location to another, basically they hitchhike a ride, and next thing you know, they're on these Hawaiian Islands and they're killing the native birds. Some of the others, so this is a zebra mussel. A lot of you have probably heard about it. Hydrilla, which is very common in a lot of the freshwater waterways of Florida. So these are all, this is by the aquarium trade. These came through shipping. So a lot of these things are hitchhikers on human vessels, and they get into places where we don't necessarily want them, or we didn't expect them to go anyway. I don't want to push it too hard because then it seems to go too far. There we go. All right, so let's talk a little bit about invasive species a little bit more specifically, and talk about the problems that they can cause. So as I mentioned, invasive species are the ones that are problematic. They're causing economic and ecological damage. So one of the things to keep in mind is they're second only to habitat destruction as a cause of local extinctions. So they're a very big problem as far as destroying a lot of local plants and animals. And if you look at this number here, the estimated annual cost just in the U.S. alone, so that's not worldwide, that's just in the U.S. alone, $137 billion every year that we spend on the regulation of invasive species. Essentially, even some of the things that I'm talking about, it's just a matter of getting rid of them from the hulls of ships or from intake pipes for power plants and things like that. Those are things that people have to go out and do. So you have to pay those people to do that. And in some cases, you're talking about with plants, the use of pesticides, things like that. So every year we're spending a ton of money, not just in the United States, but worldwide. So that's really what we're talking about. And Florida is terrible about this. As far as it is a location, not only do people like me from up north end up moving here at some point in our life. I'm originally from Nebraska, but organisms from all around the world end up migrating to Florida and getting introduced down here. Florida is a very welcoming habitat for them in a lot of ways. So because it's subtropical and tropical in some areas, basically there's a lot of organisms that are capable of living in this kind of environment. Because of the severe habitat destruction that we've had around the state, there's a lot of native organisms that aren't in very good shape, so there's some that leaves kind of spaces open for introduced species to take hold. There's lots of ports, right? We get lots of air traffic, we get lots of shipping traffic throughout the state, so there's lots of ways for organisms to get here. And then once they get here, all the lakes, the rivers, basically it makes it easy for them to move around. All right, so Florida is the number one state. It and California essentially are the two places where you get the most introductions in the United States. And if you look at, this is the most recent data I could find actually, which is way back, in year 2000, but over $90 million, almost $91 million spent on control of introduced species. Now a lot of these are things that you're probably familiar with, right? Citrus canker, Mediterranean fruit flies, navigation problems like hydrilla, hyacinth, alligator weed, those kind of things take a lot of effort to try to control. And so that's where a lot of that money is going. Now, some of the things that we're going to talk about today are some of the coastal invaders. So some of these you may be familiar with if you get out around this area at all. One of the more recent ones is the Titan Acorn barnacle shown there. So those are big, just called pink barnacles most of the time, but they can get this big around compared to the little tiny ones that you see on the native ones around here. The green mussel we're going to focus on today. Sometimes they're not quite that vibrant green. Sometimes they're even more so. They're very, in some cases, you can get a really, really almost fluorescent green color to their edge. So really pretty mussel, but they don't belong here. And then the charu mussel. This is the most the oldest introduction of these three species. They've been around about 30 years now, much smaller than the green mussel, and I'm not going to focus on them today, but there's some colleagues of mine at Central Florida that are working primarily on those. Now this is the other part of the story that we also want to talk about, because introduced species, so the question is, are they always bad? So I said that you've got introduced species and then you've got invasives that are bad. So what proportion are typically bad? Which ones become problematic? So obviously some are beneficial because we've talked about the introduction of organisms for agriculture and things like that, and also we use them in some cases to regulate other species. One of the things to keep in mind that you oftentimes hear in the introduction literature is what's called the rule of tens. And the rule of tens basically says that out of all the species that get introduced, only about 10% of them actually will establish a population locally. So most of them, they'll get introduced, they might be there for a short period of time, maybe weeks, months, few years, and then they're gone. They go away, they never really establish a working population at that location. So most of them, nothing ever happens to, and they're gone fairly quickly. Of the 10% that become established, only about 10% of those ever really cause any kind of real problem. So only about 1% of all introductions end up being invaders, essentially, or invasive, what we would call that. So therefore, obviously, the majority of them we're never really going to have any issues with, or unlikely to have issues with. So then the question becomes, can we identify which ones are going to be bad, right? So we know that they're not always bad, but are there certain characteristics that the successful invaders have? So who do we know that we should look out for, essentially? Because one of the things that you also have to keep in mind with introductions is that the earlier you catch it, the more likely you are going to be successful at controlling it. Once they become established, and once they really get their foothold, it's almost impossible to get rid of them. And it's going to cost millions and billions of dollars to try. So that's the one thing, is that it would be nice if you could identify the potential problems early. Well, here's the bad answer, is that we really haven't figured out a way to identify them. Because there's been a lot of people that have thought of different things. Well, maybe we need to look at how much genetic variation there is. So maybe if they're highly variable, they're more likely to succeed. Nope, doesn't seem to matter. There are successful invaders that have lots of genetic variation. There are successful invaders that have very little. Maybe they're generalists, so they can occupy a wide variety of habitats. Or maybe the specialists are better because that means that they can outcompete the other things in the area. Nope, there's examples of both. Doesn't seem to matter. Reproductive patterns, as far as, there's some that have really slow reproductive rates that are successful. There are some with really high reproductive rates that are successful. So none of those things seem to relate to whether or not an organism is going to become invasive, or even their ability to establish a population. The only thing that we've found that's a predictor of establishment is this last factor here, what's called propagule pressure. Propagule pressure is a fancy term that basically just means how many of them are getting introduced and how often. So the bottom line is if a lot of individuals get introduced, or they get introduced repeatedly to the same area, chances go up that they're going to establish a population. So this is really the only thing that we can do effectively, is to try to deal with propagule pressure. How can we limit the number that are getting introduced, or the number of introductions? That's really the key. So again, when you look at what can be done about introduced species, we've really got a few things that we can try. Number one is attempt to eliminate or limit the number of introductions, so cut down on that propagule pressure. So again, of some of these things. When you go on a commuter flight, they don't want you to have apples or any kind of food items with you. When it comes to shipping, they like to have the boats release their ballast water outside of the port. So those kinds of things are basically factors that you use to limit introductions. But then once they get here, again, it's usually costly to remove them, physical removal or harvest of organisms, or introducing natural enemies. Harvest can be effective some kinds, and if you're lucky, if you're using like largemouth bass as an example, there's a sport fishery associated with that. So people you don't have to pay people to go out and fish for them. They fish for them, and you just say, you have to keep them. You cannot release them. You have to kill everyone that you catch. Mm-hmm. So that can be somewhat effective, and it's cost effective. But if you don't have a fishery like that, then basically you have to pay people to go out and actually remove them. That's going to cost a lot of money. Introducing natural enemies. We've done that in a number of cases, but here the problem is you've got to be very careful, because here you're introducing a new species to try to control a previously introduced species. So if things go wrong, you've just given yourself another problem. Some of the earliest examples of those have not been good, but we've been getting better lately. Let's put it that way. A lot more work has gone into making sure that the thing that you're introducing is going to focus on what you want it to focus on, and not find something else tasty that's native and we want to keep. So really what we're going to talk about today is kind of this first aspect of eliminating introductions, or trying to limit them anyway, in relation to the green mussel that we talked about here. Now, when you start talking about eliminating or reducing introductions, there's some information, it's easy to say, oh, we just need to keep them from coming here. But obviously, that's easier said than done, because in order to keep them from coming here, first of all, we have to know where they came from and how they got here, right? So if you know those modes of transportations and the locations of origin, then you could potentially focus on where the problem areas lie, and you might be able to cut those avenues off. But then there's the second question of, if once they get here, where are they likely to go? What are the habitats and what are the areas that they most likely are going to expand to? Which ones are in danger of potentially getting these? So they may not be there now, but maybe in 5 years, 10 years, 20 years, if they expand out, where are they going to likely end up? So that way you can kind of try to cut off those areas. We know that this area is going to be a good area for these if they ever get here. So we're going to try to keep them out, okay? So essentially that's what we're going to talk about really with the green mussel. How many of you have seen green mussels around here? So, right, a few of you. So if you spend any time around, they're probably a little bit more common up in the St. Augustine Inlet, but there's some in Matanzas Inlet. I used to collect a whole bunch of them off of the dock behind the Whitney Lab. And the little area just south of there, the little marina there, if you walk around in there, you can oftentimes find green mussels in there as well. So these guys are native to the Indo-Pacific, and I'll show you a picture of their range in a little bit. They occupy low intertidal and high subtidal habitats. Like most mussels, they always grab onto something. So basically the larvae float around and then they'll settle down, and these guys settle on hard substrates, things like rocks primarily, and they'll attach to those rocks, and then the adult basically spends its whole life there. It doesn't move around very much. These guys, their larval period's about 2 weeks, so they float around in the ocean for about 2 weeks before they settle. And then once they settle, they grow quickly and they can become reproductive within 3 months. So they can settle in the beginning of the summer and they can start reproducing by the end of the summer in a tropical habitat. So what about the problems of these guys? Well, they're a known biofouler. They were originally found over in Tampa because they were clogging up the intake pipes of power plants over there. So that's when they were first found over in Tampa Bay. So every year they have to go out and unclog those pipes. They attach to ships, things like that. In Tampa, they had been suggested to displace some of the oyster reefs over there. And actually some colleagues of mine down at Central Florida showed that green mussels can negatively affect the settlement and growth of oysters. So this would qualify as being an invasive species, and that it is having some negative impacts in this area. So what we're going to do is, so if you look at the history of introductions, so this red coloration there on that map is basically showing you the native range or the historical range of the green mussel, so the Indo-Pacific, the waters of the Indo-Pacific. But in the Western Hemisphere, they've been introduced here, I guess, in the last about 25 years. So the first introduction, they were first reported in Trinidad in 1990. And shortly after that in Venezuela and Jamaica. By 1999, they've been found in Tampa Bay. And in 2002, there were some reported in St. Augustine, in the St. Augustine Inlet. In 2003, there was another report of them in waters of Georgia. Most of them are offshore on artificial reefs in Georgia, but they even found a few shells, no living animals, but they found a few shells in the waters around Savannah. So anyway, if you look here, they have been introduced to the Western Hemisphere, and it looks like they're definitely the number of introductions has been expanding, let's put it that way. So what we're going to do is really kind of address those questions that we talked about before, specifically for green mussels. So first we're going to try to figure out where green mussels in the U.S. came from. And we're going to use a genetic analysis to do that, and I'll talk more about that in a second. And then the second question is looking at whether or not green mussel populations are likely to expand beyond the borders of Florida. So we know they're here now and they're on some offshore reefs in Georgia, but are they going to sweep up the entire Atlantic coast of the United States? Are they going to get throughout the Gulf? Those kind of questions are what we're going to try to do. And basically we'll do that with development of what's called an ecological niche model, which incorporates the habitats that an organism uses, its physiological tolerances, what kind of conditions it can live in, and also how far it can disperse so that we know how quickly it can get to these kind of areas or whether it can get to those areas. So let's look at the first one, the first question of what is the source population of the mussels that we have in the United States. So again, we're going to use genetics to do this. And the easy way to think about this is like a paternity test. So if you think about a paternity test and the way that it works, you can essentially eliminate certain possible fathers from contention based on them not sharing the right genes with the offspring. And so this is essentially what we're going to do here. It's the same kind of thing where we're going to genetically analyze some of their populations within the native range, and then we're going to compare those to the populations that we have in the introduced areas. And basically the idea would be introduced populations should have a similar genetic signature as the population that they came from, because that's where they got their genes from. So we've got a couple of different possibilities here. If you remember the previous slide when I was showing you the history of introductions in the Western Hemisphere, it looks like it was kind of stepwise, right? It looks like, first they showed up in Trinidad, then they were in Venezuela, then in Jamaica, then in the US. So it looks like obviously they came from the native range to the Western Hemisphere. So one possibility would be that they were introduced to Trinidad, and then the mussels from Trinidad have been introduced to these other locations. Another possibility is that each one of those different locations, Trinidad, Jamaica, the US, has received a separate introduction from the native range. Those two possibilities give us very different predictions, because if the ones from the US came from Trinidad, then the ones in the US should genetically look like the ones in Trinidad. Whereas if there was a separate introduction from the native range to Trinidad and a different introduction to the United States, then the ones in the United States should look very different from the ones in Trinidad. All right, so that's essentially what we're going to attempt to do, is distinguish between these two potential hypotheses. All right, so if we look at, let me just kind of walk you through these data real quick. So these are what are called haplotype frequencies. Haplotypes are just different copies of the same gene that differ in some way. So I've color-coded them. So every different copy has a different color to it. And these are three populations that we sampled within the native range, so Hong Kong, China, the island of Singapore, and India. And one of the things that's very typical, the way that this can work, is that most organisms that occupy a very large geographical range oftentimes genetically differ from each other. And you can think about humans in that regard as well, right? Because if you look at a person from Africa and compare that to a person in Europe and compare that to a person in Asia, not only do we look different, but we are genetically different as well. The same thing is true of most organisms. If they're spread out over a large area, usually populations have genetic differences. And you can see that, right? Compare India to these other two. India is very distinct from those other two populations. Singapore and Hong Kong, more similar. You see a lot of the same colors there, but some colors are at higher proportions in one population than the other, and some of the colors in Hong Kong are absent in Singapore. So now we can look at the introduced populations and see which of these native populations they most likely came from, and again, does it look like they came from a single introduction, or were there multiple independent introductions? Well, the good thing about the introduced populations, if you look at those, they all look the same, right? So it looks like there was a single introduction to the Western Hemisphere, most likely to Trinidad, and then that population in Trinidad has then been expanding out or being introduced to these other locations. So we can blame Trinidad, all right? They gave us our mussels. Now, then we can ask the question, okay, well, where did they come from? Where did Trinidad get theirs from? So this is where the paternity test comes in. Because obviously you can eliminate India, because none of the colors that you see in the introduced populations are found in India. We don't see any of those, so they know they didn't come from there. Hong Kong and Singapore, we said there's more similarities to, but one of the things that you notice, look at this light red color in all the introduced populations, that it's a very high frequency in each of those populations. That color is not in Singapore at all, but it is in Hong Kong. So again, this suggests that most likely the mussels in Trinidad came from China, essentially, or maybe Hong Kong directly, but definitely somewhere in that area. So most likely went from Hong Kong to Trinidad, and then from Trinidad they've been expanding out from there. So again, if we want to use this information to regulate introductions or eliminate introductions of green mussels to this area, we've got to be looking at boats coming from the Caribbean, essentially, because these other populations, that's where we seem to be getting them from, at least for right now. All right, so again, the data support a stepwise introduction from China to Trinidad, and then from Trinidad out to these other locations. Most likely that's coming from ballast water of ships, obviously, because these are a marine organism. That's the best idea. Most likely they didn't get on an airplane. But the other thing, there's another piece of data that suggests that China is most likely the source for the mussels in Trinidad, because China ranks 5th in overall shipping imports to Trinidad. None of the other countries within the native range of the green mussel even rank in their top 12. Very little shipping from any of the other areas. So again, both the genetic data and the shipping data suggest that most likely they originally came from China and then have been expanding out since then. Now, one of the other things that you notice, if we go back real quick to hello? Shouldn't have done that. I should learn. Hello? Is it not? There we go. One of the things that you notice here, look at the number of colors. So in these populations, these introduced populations, you've only got 3 or 4 different copies of this gene, whereas in Hong Kong, Singapore, India, you've got like 14, 15 in each of those that we found, even though the sample size is similar. When you lose that much genetic variation, that suggests that you had a very small number of individuals in the original introduction. And in fact, our models suggest that the original introduction to Trinidad probably consisted of about 7 individuals, 7 reproductive individuals that survived and reproduced. So a pretty small introduction that has been successful. So again, propagular pressure is the best predictor of establishment, but even some introductions that are really small can be important. A lot of you may know the story of the lionfish here, which has kind of a similar story as far as lionfish were most likely introduced through the aquarium trade, and the estimates is that it was a very small number, about 5 to 10 individuals that were originally introduced, and now those things are everywhere, right? Okay, so now we've got the second question of where are these guys going to expand to? So the idea here is to develop what we call an ecological niche model that can identify locations that can potentially support this species, even though it isn't there yet. So this is going to depend on the physiological tolerances, so what kind of temperatures and habitats can they tolerate, what types of habitats do they like to be in, and also the dispersal capabilities. Where can they get to and how quickly? So is it going to take 2 years to get there, 5 years, 10 years? We need to look at kind of a time frame. So the first thing we're going to do is look at temperature. So temperature is oftentimes considered the most important environmental factor for a lot of these species, especially those from tropical areas. So if you look at a marine invertebrate like a mussel, temperature directly affects its physiology because its body temperature is basically the same as the temperature of the water or the air surrounding it. So everything, its growth, its reproduction, it's all dependent on temperature. Mussels, if you remember, they're what we call sessile organisms, so they sit in one place. If the temperature changes, they can't get up and move. They can't move to a warmer spot. They've either got to deal with it or die. So temperature can potentially kill them if it's a bad situation. They're also intertidal, which means that we have to factor in not only water temperature but air temperature, because parts of the days they might be exposed to the air. And again, since these are from tropical areas, it's expected or hypothesized that it's most likely cold water and not warm water that's going to be the limiting factor. So we know that they can survive in Florida, but could they tolerate wintertime temperatures north of here? That'll be the real question. So this is what a graduate student of mine worked on several years ago, basically looking at their mortality in cold water when exposed to cold water. So what she did is she had basically a control group, which is shown up here. This is just looking at the number of days they survived, up to 30, for mussels that were held at different temperatures. So 21 degrees, that's kind of so 21 degrees room temperature, about 72 or so degrees Fahrenheit. That temperature was our control, very similar to a fall temperature in a lot of these areas. But then we looked at 14 degrees, which are the gray squares there, 10 degrees, which are the black triangles, and 3 degrees, which is the circles there, the gray circles. The reasons we chose those temperatures is basically because 14-degree water is about as cold as it ever gets in the Northeast Florida area. Every once in a while it can dip down below that on extreme cold snaps, but 14 is typically the minimum temperature that is observed in the wintertime. 10 degrees is a little bit further north. That's the wintertime temperature minimums that you see in Savannah area. And then 3 degrees was kind of our extreme. And if you notice, what happens is even at 14 degrees, the temperature that it could potentially reach in Northeast Florida, basically the mussels can live up to about 10 to 14 days at that temperature. By the time you hit about 2 weeks at 14 degrees, 90% of those animals are dead. So cold snaps are definitely going to kill them. 10-degree water, they can only live maybe 7 to 10 days at most. So bottom line is cold water kills them. Same thing if you look at cold air. All right, so this is basically the same experiment, just looking at how exposure to, like if low tide was in the middle of the night and you had a cold night, that was all this was like a 3-hour exposure to cold temperature, and then looking at what happened to them after that. So after that, they were put in water again. And basically you see the same kind of thing, 14 degrees, 7 degrees, and 0 and minus 10. Those were our extreme temperatures that we did there. And every single one of them shows significant mortality of green mussels if they get to those cold water or cold air. So looks like cold is going to kill a lot of them. And we've actually seen that in the local area and in Tampa. If you have extreme cold snaps, that's actually resulted in high mortality of these guys. So we know that mortality occurs within 2 weeks at temperatures of less than 14 degrees, both water and air temperature. In this part of the country, in Northeast Florida, typically don't get that cold for very long. So what we want to see is how does this compare to where we find them now and other wintertime temperatures along the Atlantic coast. So this figure over here is showing you where we currently find green mussels in this state. So the green highlighter is showing you, again, coastal areas of Northeast Florida, around Tampa, and also some offshore sites of Georgia, which tend to be about 3 degrees warmer than they are on the coast. And that's going to be important here. So again, if you notice what winter water temperatures tend to be, in the St. Augustine, typically about the minimum temperature you're going to find is about 14 degrees. So right around where we said mortality is going to start to get bad. Up in Savannah and Charleston, you're closer to 10 degrees. So again, in Georgia, we don't find any of them along the coast. It's too cold there. Offshore, where it's about 3 degrees warmer than this, it's going to be up closer to 14 degrees. So they can handle that a little bit better, at least for short periods of time. But again, all those areas along the Georgia and South Carolina coast, we don't find them in, most likely because, at least partially, because they die in the winter. But as many of you may realize, we have severe climate change happening where the sea temperatures are increasing and have been increasing for quite some time. And most models project that the sea temperatures are going to increase about 1.5 degrees at least over the next century, and up to maybe even another 6 degrees almost in the next century. So if we look at what's going to happen, so we know that green mussels currently occupy this part of Northeast Florida because it's too cold in the winter everywhere else. But with just a 1.5 degree temperature shift, they're going to be able to occupy up through the coast of Georgia. And if the models on the extreme upper end are correct and we get an increase in temperature of about 6 degrees, in the next century, they could expand north as far as North Carolina, at least with the temperatures that they could tolerate. So as long as there's habitat that they like up there, they should be able to expand over the course of the next century.
Could you remind us what 10 degrees centigrade is?
10 degrees centigrade is about 50, low 50 Fahrenheit. Right. Okay, so the other aspect of this as far as habitat is what kind of substrates do they like to settle on? So I said that these things settle on hard substrates, so they attach to hard substrates. But are there certain things that are more attractive to them? Oftentimes these marine invertebrates, they're very specific about the kinds of places that they settle. So we need to know what they settle on because that's going to tell us how much habitat is in all of these areas that they might be able to get to. So this is actually from a different mussel. This is Mytella charowana that I mentioned before. And this was just a preliminary study done by some people at Central Florida working in the Indian River Lagoon. And what they found is when they found these Mytella charowana, these other mussels, that most of them were on man-made substrates. They're in red. So things like dock pilings is where they were finding these. Live oyster clusters tended to have quite a few, but most of the mussels they were finding were on things like docks. So this gave us the idea, what if that's going to be the case here? Because if you think of Northeast Florida, there's not a whole lot of hard substrate, right? We don't have rocky beaches. We have sandy beaches. We have these, salt marshes, things with very soft sediment. We do have docks, though, right? And we do have lots of boats, and we have some bridges and riprap associated with bridges. So if those are the kinds of substrates that these things like to settle on, then that's going to provide a lot of habitat for them. If they don't like to settle on that, then maybe it's not so bad. So we wanted to see if green mussels were going to settle on certain types of substrates more than others. So what we did, this is up in Sisters Creek in Jacksonville, is we would put out these frames that looked like this that had a choice of substrates. So we had 6 different substrates that we offered them. So 3 of them were shells, so natural substrates of other bivalves like oysters, green mussels, and Mytella shells. And then we gave them options of what we called man-made, even though wood technically isn't man-made, it's associated with docks, so that's why we called it a man-made substrate. So wood, brick, plexiglass, brick representing dock piling, or not, but bridge pilings, and riprap, plexiglass more like boats. So we gave them the options of those types of substrates to settle on. And basically we came back after a month or 2 of leaving these in the field, and they'd look like this. And we'd go through and we'd try to find green mussel spat. Spat is just it's a scientific term for a recently settled larva is all it is. So it's a little guy. So we would look for green mussel spat on each of these and figure out where they settled or which ones, what they preferred to settle on. And this is what we found. If we left them out there for a month, that most of the settlers were on shells. They didn't seem to like the man-made substrates so much. So things like dock pilings, plexiglass or wood, plexiglass and tile, they got some settlement, but not nearly as much as what we saw on the shells. So they seem to settle on shells more so than those man-made substrates. So that seems good at first. After 2 months in the field, though, if you notice what happens is it all equalizes. It evens out. So for whatever reason, they tend to settle on shells first, or when they first settle, but then you've got basically no differences among these after 2 months. What I think is going on is most likely you get so many of them on these shells that they start competing with each other and survival goes down. So eventually you end up with about the same numbers across all the substrates. So the good news is they don't seem to especially like man-made substrates, but they do fine there. So in areas where you don't have a lot of shells as options, if there's not lots of oyster reefs or something like that, then obviously you can still get settlement in those other types of habitats. All right, so then the last thing that we're going to talk about are really the dispersal and settlement patterns of these mussels, trying to figure out a couple of questions. Really, is mussel larval settlement habitat dependent? And what we're going to look at here is mostly habitat based on salinity as much as anything else. Are there certain salinity environments that it likes better than others? How far do the green mussel typically disperse? And also the best part, or the most important part, is can we predict where they're going to go? So if you can use a model of the physical oceanography, so the currents in an area, can we use those models to say, oh, they're going to travel this far this year, and then next year they're going to go this far? Because that allows us to project out for over longer periods of time. So we'll see if we can do those things. So this was all done within the GTM NER area and a little bit further south, so all parts of the intercoastal waterway. So basically what we know is that in the intercoastal waterway, there's two main locations where there's quite a few adult green mussels, right around St. Augustine Inlet and right around Matanzas Inlet. Just up here, I used to have this place, I used to call it the Magic Dock. I don't know what was about it, but there was a magic dock right up here in Matanzas Inlet that I used to be able to go and rape and pillage hundreds of mussels at will. I mean, there were just tons of them on that thing. So anyway, you know, right in Matanzas Inlet, right around St. Augustine Inlet, that's where the adults are. So we can actually take advantage of this, because if there aren't very many adults in other locations, what we can do is, so if, just as an example, if we know some adults are here in Matanzas Inlet, and you get, you know, mussel spat settling down here, you know that they had to travel at least that far to get there. So that's basically the idea that we went with, is we put these different sites out in the intercoastal waterway at different distances from the source populations, the adults, where we could look at where they settled, how many of them settled there, and you can get an idea of what the average dispersal distance is and what the maximum dispersal distance is. And the other thing that we did is we also had several locations that were in the intercoastal waterway, but we matched them with a location in a feeder creek that was about the same distance from the source population. But again, feeder creeks, salinity is going to fluctuate a lot more than within the main channel of the intercoastal. So that was the habitat aspect of it. Do we typically get settlement in the main channel where the environmental conditions are pretty constant, or are they settling up in these feeder creeks, making that habitat effective for them as well? So this is what we would do, is we would go out, we would make these little collectors, which was just a big PVC pipe that we would strap onto a channel marker out in the intercoastal. And then down at the bottom of that PVC pipe, we would have a frame with these tiles. So again, giving them a hard substrate that they can settle on. So we'd have these 4 tiles, 2 on each side, that we would put out there at each location. And then every month we'd go out, we'd pull this in, we'd collect the tiles, we'd put new tiles on there and put it back out there. This is what one looks like after it's been in the field for about 3 months over the summer. As you see, it's all encrusted with barnacles and all of these green things. Those are all green mussels there. That one was up just at the mouth of, oh man, which one? What creek is it? Oyster Creek up by St. Augustine Inlet. So anyway, we would take those tiles and we'd figure out how many mussel spat were on them. So the first question, is larval sediment habitat dependent? Do they like to stay in the main channel, or do they also like to go back into the creeks? Well, all the creeks are on the left-hand side here. So Oyster Creek, Pellicer Creek, Moses Creek, Cassacola Creek. And then these are sites that are basically the same distance, but they're in the intercoastal main channel of the intercoastal waterway. So if you look there, pretty obvious, they tend to settle in the intercoastal waterway, the main channel. They don't get back into the creeks, all right? So it seems like the highly fluctuating salinity of the creeks is not a good thing for them. The next question is, you know, how far do they go? And so this is just showing you the number of settlers that we found in each of our different locations, and this is just looking at within the intercoastal waterway. And one of the things that you notice is you tend to get a peak kind of down at this end and another peak down at this end. And basically those peaks relate to the inlets. So you get a lot of settlement right around St. Augustine Inlet, you get a lot of settlement right around Matanzas Inlet. And in fact, if you look at the number of spat that we would find related to distance, most of the settlement tended to occur within about 5 kilometers of those inlets. So they don't seem to be going very far. So that's a good thing. They aren't going to disperse too far, it doesn't seem. But we did get some settlement as far as almost 20 kilometers away from those inlets. So again, they are capable of dispersing, you know, out to 15, 20 kilometers. But again, it doesn't seem like they do that very often. So what about the modeling? So Peter Sheng is a modeler that works at the University of Florida. So we collaborated on this. So he has a physical oceanography model of this area. So it incorporates the tidal currents. So every day he knows that you give him a time, he knows what the tidal current was doing, he knows how much the wind was blowing and all that. So what we were able to do is we were able to use the settlement patterns that we saw in the previous study, and we say, okay, we know they settled on this date. So given the time that the water temperature, we know how long they were in the water column because it takes that long for them to develop. So we would give them specific time periods and say they were probably born around this date and they settled on this date. If they were launched from the St. Augustine Inlet or they were released from the Matanzas Inlet, where would they go if this model was predictive? So this is showing you this black blotch up here. There's 1,000 particles that were released into the computer, essentially, on these different dates. And this is showing you where these particles are going. So if they're released from the St. Augustine Inlet, if you notice, in this case, they all get sucked out into the Atlantic Ocean, where they go down along the coast. And this is after a week, and this is after 2 weeks when they would be close to settlement. Same thing, Matanzas, most of them get pulled out of the Matanzas Inlet and then go southward. But some of them do get down into the intercoastal waterway as well. So what we're able to do is we take those models and we say, okay, we know how many mussels we actually found in these various locations on these dates. So does that correspond to the number that we would predict would be there? So are the places where the model suggests that larvae should go, is that where we're actually finding them? No. So if you look here, so this is the model run under different conditions. So during an ebb tide, so if the larvae were released on an ebb tide, if the larvae were released on a flood tide, or during a peak high tide. And if you look down here, this is the number that we actually found, the density that we found in different locations. And this is the percent of the particles from the model that ended up there. So again, if you look in these, it's just a flat line. It doesn't matter how many we found there. The number of particles that we expected to find there was virtually none. So that's not good. Obviously, there's something wrong. Our model is not predicting what we're actually seeing. So this is really where a lot of my work right now, or some of my work is focused on, is trying to figure out why. And we'll talk about some of the ideas there in a second. But just to give you kind of a summary of this part, so we saw that green mussel settlement was habitat dependent. We didn't get much settlement in the feeder creeks. Most of them stayed in the main channel. They didn't seem to travel very far. Most settlement occurred within 10 kilometers of the source, but there was some that was observed beyond that. And again, this is the bad part, is that our model was not correlated with what we actually saw in the field. So here's the question, right? Why doesn't it predict? Well, there's a few things here. So again, when your model doesn't fit your data, typically it's because there's assumptions that you're making that aren't correct. There's information that we don't have. And that's probably what's going on here. So we have no idea the time of day that these mussels spawn. If they spawn on an incoming tide as opposed to an outgoing tide, that can change the outcome significantly. We don't know if the green mussels larvae, when they're in the water column, if they move up and down. So we know that they can't move much horizontally compared to the currents. But there are some, like blue crabs as a good example. Blue crabs migrate up and down in the water column to take advantage of incoming and outgoing tides at different times of day. So they go down to the bottom when the tide is going out, and they'll come up to the top when the tide is coming in, and they can get directional movement. We have no idea if mussel larvae do that or not. Most mussel larvae do not. We don't know if green mussel larvae do that. And I've currently got a graduate student that's exploring some of that. She was collecting basically larvae in the plankton at all different times of day on different tidal cycles, and basically looking at do larvae move according to where their food is, according to the temperature, according to the salinity, to the time of day. We're trying to look at all those different aspects to try to get a better handle on how we could maybe tweak the model to hopefully make it fit better. All right. Okay, so with that, I'm pretty much done. I think I'll just summarize a couple things if this moves. Point that way. There it is. All right, so again, just to summarize the things that we went over, we saw that the green mussels in the United States most likely came in the ballast of ships from the Caribbean islands specifically. The current range of green mussels seems to be limited by cold water in the winter. And green mussels in this area tend to settle on natural substrates in areas like the main channel of the intercoastal waterway, where the environmental parameters don't vary much on a day-to-day basis. And green mussel larvae seem to disperse about 10 to 20 kilometers, but unfortunately, as I said, we can't actually predict that based on the currents at this point in time. So with that, I'd just like to acknowledge some of the other people that worked on it. There were a number of students that were involved in these various projects. Some of the collaborators, mostly from the University of Central Florida, and Peter Sheng from University of Florida, helped out, and we had received some funding through the USDA and also some internal funding from the University of North Florida for a lot of this work. So with that, I'd like to thank you for your attention. I'd be glad to take any questions that you have. Yeah.
First, nice job. Good use of graduate students. That's what they're there for.
Right.
I used to do the same thing with human beings in amusement parks.
Yeah.
So your variance matrix must have been huge to figure out what the correlations were or were not. My question is, expansion seemed to stop in 2002 unless that's where simply your slide stops. And the other is that Tampa attracts them and St. Augustine would attract them according to the geographic. But the huge sections of Florida that would seem to fit the habitat requirement show no at all.
Mhm.
So given the chi-square and leaving that all alone, why? What has a lot of research been done on why they're not going to certain places? Because that might be more predictive for an F-test on the model.
That's a good point. And actually so the easy answer to your question is no. Not a whole lot has been looked at.
You need somebody to do it.
Yeah,
free.
exactly. Not a lot has been,
I got the PhD in the field, I can do it for you.
Right. After they were first introduced to Tampa, there was a huge expansion.
Mm-hmm.
That has not entirely quit, but it's definitely decreased over the years. Now, one thing I didn't talk about because I didn't do any work on this, but there's two things that we know that seem to be associated with high mortality events. One is cold wintertime temperatures, which won't predict why South Florida doesn't have them. But the other thing is they seem to get knocked back by red tides. So in the Gulf, red tides will kill them. So that's been a big issue with them out there. Again, they expanded out. A cold snap in northern Florida in the Gulf knocked them back, I can't remember, maybe 8 years ago or so. Populations decreased quite a bit. But red tide events also results in significant mortality. So that could be what's going on further south, is that because we do get more consistent red tides in that part of the state. So that might be a part of the issue. But definitely the physical oceanography suggests that they should predominantly move south. So we know they're here, so they should be moving south, so why aren't they establishing down there?
If, by way of follow-up, if they transport mostly by ship via Hong Kong to Trinidad, why would they not similarly, and I can understand the dispersion to Jamaica and the rest of the Caribbean because of cruise ships and other, why would they have not transported to Fort Lauderdale, Miami area, where the greatest number of ships come in on a daily basis?
Right. That's a great question, and I don't know the answer to that, other than, again, we don't know everything about the habitat that these guys like.
Right.
What we do know, they tend to not be, I'd have to look at what the tidal fluctuations are like in that part of the state. Because up here, the tidal amplitude's fairly shallow compared to a lot of places like up in Georgia. Mm-hmm. And so in Georgia, along the coast, not only do they have to deal with the low wintertime temperatures, but most of the places that they don't like being dry for very long. Let's just put it that way. So they tend to be more barely subtidal than they are intertidal. So that leaves them a lot of habitat on docks and stuff like that in this area, whereas in Georgia, when that tide goes out, they're dry for hours. So I'm thinking that might have something to do with it further south as well, because I believe the tidal amplitude around Miami is much higher than it is here. And so it's probably leaving a lot of their habitat high and dry for too long. That would be my guess, but I don't.
My wife wants to know if they go well with garlic butter.
They're a big agricultural crop in the Indo-Pacific, and that was actually, so the problem here in this area is that most of them live in areas where you can't harvest shellfish because of pollution issues. So depends on how much you want to test your immune system. But I will say that that's one of the things when I went to Jamaica to collect those. They told me, one of the things when I got down there, they were like, "Oh, you should have been here a couple years ago." I'm like, "Why?" Because there were so many more of them. You could go anywhere and find them. I was like, "Well, what happened to them?" Because I was like, "They aren't getting killed by the cold down there." And they said, "No, we think that the locals figured out that they were good to eat." And so you could go out on a lot of the beaches and you would find areas where somebody had a little campfire on the beach, and there would just be green mussel shells around them. So whether or not that's true, whether or not obviously there were people that were harvesting them. Whether or not it made that big of an impact on their populations, I can't say. But yes, you can eat them. They are tasty, evidently.
Thank you.
Yeah. Yes.
So are they a problem in their home native areas, like they are here?
No, in the native areas, they're used primarily for agriculture, and they're one of the main. They don't really have the oysters over there. They're the main bivalve in that area that they use for this kind of stuff. So they're normal over there. Yeah.
Does that lack of genetic diversity create a potential endpoint in their thriving, or could more research into that lack of genetic diversity be used to solve this problem?
Yeah, so the thing with genetic variation is that genetic variation is the source for adaptation. So whenever you get a species that moves into a new area, so one question that I would have is, so we know that green mussels in this area can't tolerate water that's colder than about 14 degrees for very long. So the expectation, that was one of the things that we actually tested is, so if you settle one year and you go through a winter and most of them die because it's too cold, then the ones that live are the tolerant ones. Mm-hmm. So when they reproduce, then the offspring should be a little bit more tolerant than what the average population was before. Mm-hmm. So one of the things that my grad student did with the project on the temperatures is we looked at whether or not the tolerance was different for older mussels compared to younger mussels. So if the older mussels were the ones that had survived the winter time, then those should be the most tolerant ones out there. And what we found is there was no difference based on size. So the oldest ones were just as tolerant as the youngest ones. So anyway, but you're right. That's the only thing that we would say is that if genetic variation is lacking like it is, then there's less of a chance that they would most likely be able to adapt to colder temperatures. But again, we also have other issues of increase in traffic to Jacksport and stuff like that, which might bring additional introductions because again, where is a lot of those ships coming from? A lot of them are coming from the Indo-Pacific. That's where most of the traffic or a lot of the traffic in Jacksport's coming from. So if we get some additional introductions, then that's going to increase the genetic diversity and who knows what's going to happen. So right now there's not much variation, but who knows what's going to happen from there. Yeah.
Has there been anyone who's looked into the artificial reefs off the coast of Florida to see if they've colonized there?
Not that I know of.
I think if they're in the reefs off of Georgia, they would have no problem establishing them.
You would think, right. So I don't know that, nobody's ever told me about them, let me put it that way. But yeah, the ones in Georgia, they initially found them on a buoy that was marking the reef. So there was a buoy out there marking the reef, and if you kind of turned up, the bottom of the buoy was just encrusted with them. So then they started looking, and they're all along the chain, and they go down to this reef. But so again, it's one of those things. It may depend on it. The temperature they can definitely handle. Whether or not the depth difference, I have no idea how depth relates in those two areas. So that might be an issue in some cases. But I imagine there's some reefs in Florida that have them. Yeah.
This is kind of a two-part question. First is, what ethically can we do as an individual to help this, or because I've seen them on St. Augustine Municipal Marina, and I grabbed them and I tried to jump them in the garbage, no one seeing.
Right.
But I don't want people going out there smashing clams on the dock and stuff like
And
that.
thinking they're a green mussel.
What can we do collectively, as in to help management issues? Like, how do we get involved here?
Well, I think that what you described is in most cases it's one of those things that makes you feel good, but it probably doesn't have a huge effect. As far as because the problem is these things. Each of these individuals, a single female releases well over 1 million eggs whenever she reproduces, and so the idea that you're going to go out there and smash a couple of green mussels and that's going to magically get rid of them, it's probably not going to do much of anything. The only thing I would say is just take care if you have a boat and you keep your boat in the water. Make sure that you keep it clear of green mussels and other fouling organisms, because that's how they're going to get transported around, more effectively than anything. Killing the few here, the best thing that can happen is we get a cold winter. I know Floridians hate to hear that, but it has affected this area as well. I mean, because there were plenty of times, years ago, where I'd go to these areas that were just, you'd find docks that were covered with green mussels. They love floating docks. And then, we have a cold snap. We had, one of those, we had like 2 winters in a row. I can't remember what years it was, like 11 and 12, something like that. We had like 2 pretty cold winters in a row. And the next year, I could hardly find a green mussel anywhere. Places where I went and there were hundreds of them, they were virtually gone. And so that's really seems to be in this area what kills the most of them. I mean, you'll never kill as many as Mother Nature will. The problem is those kinds of winters are going to get rarer and rarer, and so and we don't get very many red tides. So I think that right now they're bouncing back. These last few years have been really mild in the wintertime, and their numbers are coming back in a lot of those places where I used to find them.
So then for this species, eradication probably wouldn't be a reasonable solution. You're just looking at management then, maintain them.
I think for most introduced species, management is about all you can do, because the problem is that these guys, if you think about the waters around St. Augustine, because I've had people that fish off like the Bridge of Lions or something like that, and they'll get hung on something and it'll come loose and they bring in their line and they've got this cluster of green mussels and other stuff that they pulled off the bottom. If you think about how can you go down there and get there? It's not like the water's so clear that you can just go down diving and collect all these green mussels. So even if you tried, there's going to be some that you miss. And again, with a single female producing as many eggs as she can, they can repopulate, relatively quickly. So, it's one of those things that I think they're established here. I don't think there's much we can do about them going away. Right now, we're pretty lucky in that they're not in huge numbers. And I think there's a variety of reasons for that, but like the stuff that I showed that they can be problematic to oysters, I doubt that they're affecting oyster populations in this area, just because the number of green mussels that are around are so small compared to the number of mussels that they probably don't have a significant effect on settlement and stuff like that. It's only when they get at higher densities you're going to see that effect. So, if they continue to expand and increase in numbers and they aren't getting knocked back by consistent, cold winters, then they could become more problematic. But right now, really, I think like the FWC and everything is looking at them more of, okay, they're here, they don't seem to be causing too many problems, they're bad, they're not real bad. Because when they first got introduced, they were throwing money at this stuff left and right. They were like, oh man, these are going to be the marine version of the zebra mussel. They're going to get everywhere and they're going to cause all kinds of problems. And they haven't done that yet. It hasn't happened. Because like I said, they've gotten knocked back a couple of times. But whether or not, the other issue with introduced species is a lot of them have what we call a lag time. There's a lag period where they get introduced and they kind of hang out in an area and they're in relatively low numbers for years, and then all of a sudden, just boom, they spike and they're problematic. You know, it's not like kudzu was just immediately a problem. You know, so these kind of things, oftentimes you have this lag period where it looks like nothing major is happening, and then the next thing you know, it's an issue, and by that time it's too late. You know, so anyway, like I said, they're here. It's tough to get rid of them.
You talked about your collection methods.
Mm-hmm.
What's the, do you need to ask permission if you're going to private floating docks that are owned by somebody? Do you need to talk with each landowner?
I normally didn't use private areas for that, but in cases where I did, yeah. I mean, like one of the places that we collected was off the Conch House Marina up in St. Augustine. So I talked to them and made sure that we got permission, and we would just go and we'd hang a collector off of one of their docks. And so yeah, you know, normally I'll go down there and I did that. Oh, right over here at you know, in Matanzas Inlet. Nobody was home, so I took a swim out. I didn't walk on their dock. I took a swim out to their dock because I looked out there and I swore I saw a mussel on it, and I was like, that looks right. So I swam out there. And I was like, yep, that's it. Oh my God! And I like looked under, and there's just hundreds of them, and they were all like this. They were just massive. So it was but then, you know, cold weather came in. And that dock's not magical anymore. Yeah.
I was just wondering, so if we have additional questions, how might we get in touch with you?
You can email me if you look at the UNF biology website. I mean, it's real easy if you remember my name. It's mgilg, so my first initial and my last name, mgilg@unf.edu. That's the easiest way to contact me. But you know, if you can't remember that, just look me up on the website and of the UNF biology department, and you can shoot me a question that way. My phone number's there too. Now go enjoy the rest of this day. It's gorgeous.